Just one little disclaimer, I'm not speaking on behalf of the entire agency by any means, although this is a organized, what's called a speakers bureau event. It's meant more as an educational discussion and not trying to present any data or anything that's trying to, I'm not trying to represent the agency. So a little bit on my background. So I grew up in the military, right? So I've traveled and lived all over the world until I was like 18. Growing up, spaceflight, astronauts, I was that type of geeky kid. That was my thing, looking up to astronauts as heroes type deal. But as I became more as an adult, I got seriously into music and Afrofuturism. And it's just sort of interesting that eventually, once I hit grad school, was lucky enough to get connected to be able to learn more about some of the activities in spaceflight biology. And it's just sort of amazing that I could take a couple of my passions from my personal life and bring them towards sort of my professional scientific life. And after doing exercise physiology was my grad work, and my grad work was on bone biology, looking at, we'll talk about it later, but trying to help advance, push the needle on and helping ensure bone biology and health for deeper space missions. And then I taught human anatomy at San Jose State for several years. And after the bone biology work and that, I stayed on and kept doing some research looking at female reproductive health, which really is tied to endocrinology and overall women's health and female health. And then after that, I had the opportunity to work on a project connected to the International Space Station. And this project is called the Rodent Research Project, which has these animal habitats that you see here on the bottom, where they send mice to the ISS, and different biological experiments can be done to understand a ton of different ways that we can understand biology that we can't do to the same level with humans. Then I after leaving San Jose State, began getting more involved in data science, as well as our biorepository of specimens that after spaceflight missions, there's a lot of surplus non-human biospecimens that are still made available for scientists to continue to do assays on and that type thing. And some, a lot of the different elements of this page I'll come back to later. And that's right, I would be remiss if I didn't mention the 60th anniversary earlier this week of Yuri Gagarin's first flight, right? Is it April? Was it 12th, 1961? It was an only 108-minute mission, but my goodness, what 108 minutes it was. And there's a bunch of just amazing quotes just for having what it was, like the first human in space, and you get a pretty cool shot of what the Vostok 1 looks like. This is from a computer game type thing called Orbital Spaceflight Simulator, and here you can see the Vostok 1 orbiter over the Suez Canal and the Nile Delta. And I always got a kick because Yuri didn't control the spacecraft down, and it had like a soft landing or anything but as he parachuted down, he landed in a field, and some different farmers were nearby. And as he was going by them, this is now this individual is now an adult, but she was a little girl, and she remembered Granny asking him, "Where are you from?" He said, "Oh, I just came on a ship." And she said, "Well, there's no sea here." And he's like, "Well, I came from the sky." And it's sort of a funny little anecdote connected to Yuri Gagarin post-flight. And sort of keeping along those lines, I think science fiction has been a critical enabler, sort of pushing and helping us dream big and dream about a future that's for everyone in space, and a dream that's for a future towards bettering of society and space and freedom and humanity. And it's interesting to think about the impact of Jules Verne. He did like Around the World in 80 Days. It's a pretty old book, and he did another book called From Earth to the Moon. And there's a statue of Jules Verne in Nizhny Novgorod, which is out in Russia. And it's just amazing if you look at all these pioneers, scientific pioneers of spaceflight and explorers of others, from was it Robert Byrd that went to the South Pole, to Goddard, Gagarin himself, number of different astronauts, even Jacques Cousteau, all credited From Earth to the Moon as some of their inspiration. And it sort of reminds me about how many colleagues who are maybe from a little bit older generation than mine, and the impact that they had on Star Trek, on them to get into science, to get into thinking about space. And so a lot of what we're involved with in space biology is trying to push the boundary to get us to the moon, to understand things like radiation, to understand microgravity, so that we can go beyond, deeper into space, to Mars and beyond. So I saw that, I looked at that syllabus that y'all have, and I saw that the paper that we put out in November was one of the readings for this past week. And so really happy with especially how these graphics turned out from the review article, which sort of gives you a sense of how experiments are conducted on several different levels, right? So it's not just human experiments and evidence, as critically valuable as that is, but we can help fill in a lot of the molecular and micro and cellular and tissue level biology by using non-human animals, using cell culture. There's a lot of new technology called cells on a chip, even plant biology we've been trying to understand. And I thought this graphic on the right does a good way to sort of understand how it's an iterative process between, because NASA really has defined these, what they call the hazards of spaceflight. That is, altered gravity fields, various different types and energy and particle and doses of radiation, hostile enclosed environments, which tends to be seen about infections, because there is more propensity to develop infections when humans go to space. Confinement, sort of long-term staying in the same room for months to a year, also on psychological, but also fitness, different parameters of health. And then lastly, distance from Earth itself is a hazard, because you're no longer nearby evac or any type of expansive medical capability to support human health. And so these are and you can sort of see below, between the various analyses and experimentation and developing countermeasures, it's all part of the entire same team effort, both within NASA and the broader science community, to push the envelope, to sort of twist the cap, step by step, to develop more evidence base, so we can develop the systems that can protect human health for deep space. Just a little bit of just being conscious of the levels, the hierarchies of scientific research, from a molecular level to a cell level, tissue, organ, system, to the whole organism. Just another little reminder that as we do basic research, which oftentimes are ground tests, we then have to do either translational or even flight validation work that then can lead to human research that can lead to applied outcomes to ensure human health. So just another little snapshot of some of the other considerations is, I've always thought the time course of a lot of biological dysfunctions was fascinating. That is, it all just doesn't happen immediately as you start to get to microgravity. It was well established early on that your inner ear, there's like the utricle and the saccule. It's these structures that are involved with these little cells, the otoliths, and there's a dysfunction that occurs in this, what's called the vestibular system, which gives you an understanding of gravity and also acceleration. And that's one of the first systems that tends to be, that has an almost immediate dysfunction with shorter term space flight. And it's one of the interesting reasons that vision changes only really became understood in the past decade. Like, we don't really have the mechanism of action yet, but because there weren't missions longer than a month or two, in earlier shuttle missions, it only once we had months to 6 months to a year missions did this continual vision challenge keep arising. And so it's just sort of interesting to think about the point at adaptation and the point of dysfunction that occurs. And yeah, there's just a whole host of different alterations that occur connected to spaceflight. So if you have any other interests, like I know, like in grad school, you might be flipping through the literature, trying to find an interesting paper to include or review or write about or whatever. These are some options of some paths to learn more of like the big picture. This is pretty much just a whole big old book here at the top, which is a sort of a summary of, was it 1965 to earlier there in this decade, of all the different spaceflight experiments that were done through the space biology program. At the bottom, there's another NASA program called the Human Research Program, and they regularly produce what are called evidence reports, pretty large, nice literature reviews, in effect, that sort of help sort of give an understanding of where we're at in addressing those various hazards we were talking about. And smack in the middle, Congress puts out a commissioned survey through the National Academies, which is an extramural to NASA that puts together a academic committee that puts together sort of the next decade priorities for spaceflight biology and physical sciences, so chemistry, physics type stuff. And there's actually a new decadal survey that's being started right now. So this one is from 2011, so we should have a new one. I think they're going to publish it 2022, 2023. And so that's exciting. So what have I been involved in? So yeah, was involved with my bone biology research, which is what I'll be talking about after this slide, but a little bit on the female reproductive health. I might talk a little bit about the artificial gravity, because I just think it's such a neat thing to discuss. You know, I was so excited and honored to be part of the science team for the rodent research project when we sent mice to the space station, looking at various different, well, they had very specific science focuses per mission. And then I'll get a little bit towards some of the data science and reuse and tissue stuff towards the end. And this is a pretty good demonstration of why I believe that rodents are such a useful model organism, so we can understand the various mechanisms of action, where it would take a human months to decades to lose this type of trabecular cancellous tissue in the bone. In rodents, it takes a matter of days to weeks. And it's just a useful way to help us understand different changes that occur. And although, you know, the gold standard still at the moment is that there's about 1 to 1.5% of bone loss occurring in the load-bearing structure, most notably in the hip and the lumbar, maybe even in the femur and in the femoral neck, if I remember correctly, in humans. And when you begin to extrapolate that, that becomes serious, right? Because if that's per month, and if they're doing a several-month mission to Mars plus the return, you could talk about anywhere from 10 to 20%, depending on the duration of loss of bone loss, which is wildly significant. And the trabeculae don't grow back the same way. Once you lose that sort of lattice structure of the trabecular cancellous structure in the bone, it doesn't grow back the same. So the growth pattern is different, which is impactful. And I saw another one of the articles y'all were looking at. I didn't put the recumbent bike, the bicycle in here, but I always thought these GIFs were really useful to sort of demonstrate how the advanced resistive exercise device works. Now, this was developed at Houston. Johnson is the home of spaceflight, of human spaceflight. And this was developed, you know, and implemented through Johnson. And although one of my personal heroes on the top right is Bagley, you'll see her demonstrating how the various connections to the ARED can be augmented so that you can do a whole bunch of different exercises just by moving around the bar to different angles and stuff. And this machine is just, it's simplistically beautiful in how they designed it, because with the absence or near absence of gravity in that sort of free fall motion in low Earth orbit, they developed a vacuum system, piston system, so that, and then here's the vacuum piston system here, and to change the force that you're producing, you actually just move the fulcrum point, and that alters the force being produced on the device. It's an elegant way to work through this. And why is this so important? There's just so many benefits. I'm sure this class is much better versed at the moment than I am on so many systems in the body. From one, that a lot of the cardiovascular work is interesting. So if you don't do a lot of cardiovascular exercise in space, a lot of the fluid tends to accumulate near the heart. And if I remember correctly, some of the mechanoreceptors and the parasympathetic nervous system dysfunction actually leads to a lack of red blood cell production. Pretty much your body begins to think, oh, I have enough red blood cells. There's all this fluid accumulating near the heart. And so red blood cells production can actually fall off. And so that's just one example of the various reasons to continue to have pretty strenuous exercise in space. And so this was sort of one of the big topics of my grad work, my grad study. And so we wanted to understand, if we have bone loss in space, what happens when you have two countermeasures with that bone loss at the same time? And we used an animal model. And so an analogous to exercise, it's called cyclical loading. I'll talk about it, but we took the tibias of mice, and on their right tibia, when they were asleep, they were anesthetized. We induced cyclical compressions on their knee and on their calcaneus, and induced cyclical loading, which induced osteoblast bone formation. And then we also gave to our animal cohorts that got the drug it's called zoledronate, which is a osteoporosis drug that's part of the bisphosphonate class. And so here's a little bit of some of the hypotheses that we developed and thought through. And here's the independent variables. So you can sort of see here the calcaneus in the knee and the tibia in the middle. And while it was just on the right limb, their left limb had no compressions. And so it was a within subject control to maintain that. And then we had hind limb loading for the bone loss itself. We pretty much is like a disuse model of the hind limbs of the animals. And over the course of 3 weeks, we applied these cyclical compressions 3 times a week. And before the experiment began, we gave the drug without to the proper cohorts of animals. So a group with the hind limb loading and those who were normally ambulating. We had a group with this zoledronate drug and those who only got like a saline injection, a vehicle, and then with or without the exercise, so that it was within subject control within the animal. And after the experiment was conducted, we used a micro-CT, microcomputed tomography, to understand the bone structure, both on the cancellous tissue and the cortical tissue. And then after doing slicing of the tissues, it's a part of histology, it's called histomorphometry. It gives you an understanding of the osteoclasts and osteoblasts, and either is there bone formation, is there mineral apopposition occurring, and what's the activity of the osteoclast surface as well. And then we also did mechanical testing, where we actually took the femur and did a 3-point bend test to understand various material parameters to understand the mechanical health. I'm just going to show one bit of the results just to move on, but this is a measurement called bone volume to total volume. It's called BVTV, and it's a measurement of the tissue in the tibia. And we did have what's called a main effect across the board for our 3 independent variables. For exercise was effective at, you can sort of see it here, exercise was effective at inducing and maintaining bone. Hind limb unloading should have reduced the bone. You can sort of see the pound mark for the gray versus the black here. And then zoledronate itself was effective. You can sort of, I forgot what that symbol is called. Phi or something like that, one of them Greek symbols, right? But you can sort of see the comparison from here to there. That also had a main effect at reducing the amount of bone loss. Although there was a blunting, which was interesting, that if you hadn't given the animal the drug, the drug actually rendered the exercise less effective, right? You can sort of see that there's an 18% increase, and then over on this side, there's only a 7% increase. So there was a blunted effect of formation from the drug. So anyhow, that was good stuff. On the exercise devices, gosh, I wanted to prepare a little bit more for today, but I didn't quite get to it because this slide's about a year old. But one of the big things is, okay, the ARED's amazing, right? But that thing is huge, and it weighs at least a ton. It might even be like closer to 3,000 pounds. And so for the last, I wonder if they've made a decision. It'd be fun to actually look up, because there's been a competition pretty much between teams of engineers and exercise physiologists who are trying to figure out the most effective way to develop this device. The size of a shoebox, though, it has to have as equal as possible both the concentric and eccentric loading, and it has to be constant throughout the range of motion. And at the flick of a switch, it should be able to do more cardiovascular, think row machine, or it should be able to produce up to like 500 to 600 pounds of force. And there's power requirements and vibration requirements, because you can't vibrate the spacecraft, or that could be a big problem. So there's a couple devices that have been competing. I just love this an acronym. ROCKY stands for Resistive Overload Concentric Kinetic Yo-Yo Device. So just something to sort of pique your interest, perhaps, if you want to get into it. I was really passionate about our female reproductive research. There's just a real lack in the field of research to understand spaceflight effects. And while yes, female reproductive health does have procreation as a component, the other component here is women's health, right? And understanding estrogen and progesterone and just general circulatory endocrinology. And there is some evidence that the ovaries The corpus lutea were unhealthy in some of the earlier flight missions. There was like atresia within the ovary of the corpus luteum, which was responsible for the predominant estrogen secretion. So there's a paper just out in the last couple of weeks, finally pushing this a little bit further. And this is where I sort of began to get involved in data science, because we took a lot of the cytology that we took from it's the estrus cycle measurement of where the animals are at in their cycle. It's analogous to the menstrual cycle, right? And you can actually do a lavage and get cells from the animals. And you can actually, when you do cytology, you can sort of see the cells at their different stages, where they're at. Some of them are becoming more apoptotic. There's also different white blood cells that show up at different time points, or proestrus, where they're actually full epithelial cells. So we took this, but it took months and months and months and months in cytology to do this, and just a slow, arduous process. So we developed, when we got a student who was a computer science student at San Jose State, and we put together a deep learning model that could, so we could classify the stages more effectively using a machine learning approach, which was novel and pretty cool and fun. And I'm not going to get too deep into the artificial gravity stuff we were involved in, but it's just something fun to think about towards the future of either having like the whole spacecraft either rotating to create the artificial gravity. I think it was like Gemini 8, that one of the early 60s missions, where they actually did have this cable device, and they actually did create artificial gravity for short bursts of time in space of the whole spacecraft, which is pretty interesting. There's a couple other groups that are looking at modules on these spacecraft that could create the artificial gravity. Like, you can even see the recumbent bike in here. I think that's what Larry Young at MIT was saying, was that we could actually have a module that would be your exercise area, and you could have it give you artificial gravity while you do your exercise. And I'm a preposterously fanboy of this show called The Expanse, which does an amazing job at following the scientific rivers of microgravity, of radiation, of all the various different effects. And pretty much our work was involved in how much artificial gravity is needed to rescue a system. If you were exposed to artificial gravity for, let's say, 2 hours a day, is that enough to counteract bone loss? Or if you had it for every other day, how would that affect circulation? It's sort of the question is how much of a dose would be required for artificial gravity to be effective for various systems. So another thing that's just worth mentioning, because it's such a big deal for spaceflight, is astronauts. And that's pretty accurate. Most astronauts are coming back with some degree of being farsighted. Back to anatomy, and when you have the globe, the pinpoint of where light focuses on the back of your eyes so that things appear clear in your field of vision is called the fovea centralis. You can sort of see it pinpointed here, here, and here. But astronauts are coming back with their globe being flattened, right? So they're actually showing up with this hyperopia. Now the pinpoint is no longer at the precise point of the fovea centralis, which is the place where there's only cones. There's actually now been pushed forward. So there's some vision decrements that are occurring. There's different edema issues with swelling. There's these little bright whitish yellow spots occurring on the retina called cotton wool spots, and issues with the choroid. There's a whole bunch of different things. And we don't have good in-flight measurements to really understand this, because one of the big driving hypotheses has been intracranial pressure of the cerebral spinal fluid, because for sure there's a fluid shift that occurs with space, fluid going towards the head. And there's some other interesting brain research that looked at either sort of crowding of sections or sort of expanding displacement of sections in the brain, which I found fascinating. And there was this huge paper that came out last year. Serena Onion Chancellor, one of the astronauts who's also an MD, I think it was on a routine. It was a monitoring experiment they were doing. They actually found a venous thrombosis, so almost like there was an occlusion of the blood occurring in the jugular vein that was actually blocking the flow back down to the superior vena cava in the heart. So there are some drainage issues that are going on that are very important to understand and probably fit into some of these. And it's probably a multifactorial problem. Most art, literature I've seen makes points to there not probably being one single bullet, one of which is that it at least as far as a couple months ago, no female astronauts have presented with this far-sighted issue. So there could be maybe a predisposition for males to develop this and maybe not females, that we're not quite sure. There's not a lot of N, right? When you have astronauts, you're only talking about a certain number. I'm going a little too slow. Here's a couple papers. I thought I would just point your way. I thought the one on the left was just awfully interesting. So it takes the twin study 1-year mission. It also looks at various different 6-month astronauts. And it also includes a couple Everest climbers. And the 2 Everest climbers both had twins on the ground as controls. And using blood draws and blood data, they were looking at telomeres. And telomeres are the caps on on the ends of your chromosomes that tend to get shorter as we age. And we're finding that in space, they were getting longer, but going back to being shorter after the flight. And some of the markers of oxidative stress, and there's another piece of evidence of double strand breaks. So it's like DNA damage that's occurring, and that's what persistent inversions is pointing towards. And so it was just a really interesting, very extreme environment, right? Comparing ISS astronauts to Everest mountain climbers. And then I had a good friend, and I didn't quite work on this study, but the same lab area I was in, they took mice on this other paper, and they used the dried plum diet as a radiation countermeasure, because there had been other evidence in the literature of something about dried plum diet, being protective against radiation. So that was a interesting study to sort of consider for nutrition. I'm at 30s. I'm going to go a little bit. Let me just double check. Oh, I guess I'm not terribly good. Oh, we started 5 after too. All right. Yeah, there's a number of different repositories around the world of data. We're seeing towards the future, more and more an opportunity is going to be for data reuse, so that after the original study is completed, the data from those experiments and pubs can actually get stored in a manner so there's good metadata. There's obviously hopefully well organized data sets, and then we can capture them so that we can preserve them so they can get reused, hopefully towards machine learning, maybe even knowledge graph type graph theory based usages, so we can begin to do different spaceflight health prediction risks and other different actions. There's also a number of different biospecimen repositories. So I've been, how I actually met Dr. Bagley was through, discussing, looking at tendons, ligaments, muscle that are still actually stored in our biorepository -80 freezers here at Ames, and hopefully getting those used, and because they're available for the science public to request for further examine. You think about the science return on this, right? You spend so much money to send these experiments to space. You want every bit of science return on that investment as possible. So this is just a little bit more about the biorepository, that after flight or some of our ground experiments, they're stored in a repository, then used for various different other publications, right? Like the RR6, the Rodent Research 6, its main mission was the muscles, trying to have a formoterol, which is a beta agonist drug to block muscle loss. But from that same animals, there was feces left over. And a group at Northwestern did an amazing paper looking at microbiome changes associated with that, right? So just an example. The other group I work with really closely is called GeneLab, and it's predominantly an omics repository on either the genomic, the transcriptomic, the proteomic, metabolomic, and a little bit of methylation data as well. And this data then, as it gets reused, can be brought into systems biology and bioinformatic approaches to look at across missions and across the different molecular spectrum, various different drivers that are affecting spaceflight. And my other hat is the non-omics data. So this is the micro CT data, the histology data, if we have muscle fiber typing data, behavioral video data, it could be glucose data from the animals, all data connected to non-human research we collect after experiments, and then make that re-available to the public. I'm going to skip through this. It's new, but GeneLab and ALSDA have joined recently. And yeah, I was telling Dr. Bagley before the talk began that one of the publications that got a good bit of press in that November package of papers in November was one around the mitochondrial dysfunction, which was seen pretty much across several different several different species, and including humans, and it was the largest amount of astronaut data ever in one paper. And this is a good example because this paper probably took a year to 2 years to develop. And so one of the other big things we're trying to do is not just basic science or applied science or systems biology, but is try to leverage modeling and artificial intelligence and machine learning so we can develop operational research, so we can do spaceflight health prediction, especially as we get more and more onboard genetics capacity and sequencing capacity. A lot of this can also support autonomous health and precision medicine that can support the flight medical officers. They have as best support as possible for them to make an onboard diagnosis. So this is another area of our research, which led to a collaboration with UC San Francisco last year, where we took spaceflight data from thymus, spleen, and liver from several different animals from different missions. And we did a gene expression transcriptomic and meta-analysis of those, the most notable fold change of those genes. Those genes then became homologs that paired up with human genes in this machine learning database of databases. And then it began, through using other machine learning techniques, predicted several different associated differences that were occurring in those animals, which was just remarkable that it had the power to do that. So we have great hope that this will lead towards sort of a new direction to build in a new field. So one of the things I've been working with Dr. Bagley again on is there is these what we call analysis working groups, which have been built around our data systems to help build our systems so that they're user-friendly and accessible and can be used to enhance more and more science. And so a lot of those publications came because of these analysis working groups. And there is a new program at San Jose State where a bioinformatics course is actually part of their curriculum is being taught using spaceflight biology data. How cool is that, right? From GeneLab. So yeah, so I know this is a little, obviously this is a grad course, but if you know of any high school students, GeneLab does have a regular what's called GeneLab for high school, and students who are U.S. citizens can apply. There are a couple age and different other qualifications and eligibility, but it's a remarkable opportunity to get high school experience at this level, to get your feet wet in space bio research. And there's even another one for even younger, for this Genes for Space. And lastly, there also is an internship program called SLSTP, Space Life Science Training Program, which brings in cohorts of interns every summer into space biology. And there's a broader NASA internship for all internships available as well. And here's what I was talking about. It's called GeneLab for college, GeneLab for university. And we're hoping to expand this beyond San Jose State to other universities in the future as well for bioinformatics, space biology analysis. So I really appreciate your time. One thing I've learned getting involved in science last decade was it's inherently a collaborative team effort, especially as our teams become more interdisciplinary and interdimensional. It becomes even more important for collaboration. So thank you.